A history of microbiology. A history of microbiology is a history of medicine. It is a history of science. It is a history of mankind. It also will give you an appreciation of how far we have advanced in approximately 150 years since Pasteur first proved that bacteria can cause disease and contaminate materials. Early microbiology. A man named Varro suggested that diseases are caused by invisible organisms. Everyone thought he was crazy. But really, who was Varro? Varro was a Roman. He lived from 116 to 27 BC. He was way ahead of his time. Nothing much happened in history towards the understanding of microbiology until 1546. And a gentleman named Fracastorius, using the example of the French disease, described how diseases are spread. Here's a picture of Fracastorius. First, he said they can be spread through the air. Then he said they could be spread by direct contact with diseased individuals. And third, they could be spread by objects that people have touched. And these are known as fomites. So what was the French disease? Well, I'll ask you in class. The French didn't claim the disease. They said it was the Italian disease. And the Italians and everybody else said it was a French disease. So it's very interesting. The first microscopes weren't invented until 1590, and they were very crude. In 1665, Robert Hooke, drew a picture of what he called was a cell. But where did this name come from? This is the picture he drew. These were cells of cork. In the early 1600s and 1700s, there was a huge debate over a theory called spontaneous generation. What did spontaneous generation say? It was a hypothesis that living organisms arise from non-living matter. So according to spontaneous generation, there was a vital force that gave life its form. And this vital force could be given to non-living matter. So where did they come up with the idea of this? Well, several people observed that if you had piles of grain or piles of hay, all of a sudden mice would start appearing. And they thought that these mice actually sprang or were created from the dead plant material, the grain or the hay. They also noticed that if you left meat around for a very long time, that maggots would appear. And they thought these organisms sprang from the dead meat. They didn't realize they were fly eggs. The alternative hypothesis was that living organisms arrive only from pre-existing life, and this theory is called biogenesis. Here's an expert on spontaneous generation, that is, bringing dead matter back to life. Yeah, you all may recognize this from the movie Young Frankenstein. There were several key events that led up to the disproving of spontaneous generation. First of all, in 1688, Redi performed experiments with meat and maggots to disprove spontaneous generation. However, in his experiment, the only way he could prevent the maggots from being appearing on the meat was to put a glass plate over a jar with the meat inside, where flies could not get to it. His critics said that by excluding the air, that this excluded a vital force which was necessary for creating life. In 1745, Needham noticed that if he boiled broth and let it cool off, that organisms would grow in it. and it would become contaminated and cloudy. In 1765, Spallanzani noted that organisms from the air could contaminate Needham's broth. Sealed and boiled, the broth did not become contaminated. However, again, the proponents of spontaneous generation said that in these sealed flasks, air was excluded, and air was a necessary vital force that all living things had to have in order to exist. So both Redi and Spallanzani were not able to disprove the theory because they could not prevent contamination and also allow air to enter their sterile chambers. A couple of other side events occurred in the late 1700s. In 1772, Scheele discovered oxygen by heating nitrates. Priestley recognized the importance of oxygen for all living things, and he deemed this as one vital force. And this was the chemical, or the component of air, that contained the vital force necessary for all living things. And therefore, if you excluded air, you excluded this vital force. So, both Redi and Spallanzani experiments were criticized for not having enough oxygen present to support life. So the belief in spontaneous generation continued. In 1676, Leeuwenhoek perfected the first compound lenses and makes a microscope that has the ability to magnify objects 300-fold. Here's a picture of Leeuwenhoek. Here is his microscope. This is a picture of Leeuwenhoek using his microscope to observe organisms in a drop of rain. The only source of light he had, of course, was sunlight. So that's why he's looking up at the window. Leeuwenhoek was able to identify microscopic organisms as being too small for the human eye to see, but there was still no connection between these living creatures and disease. In 1796, Jenner performed the first vaccination experiments against smallpox. He developed a vaccine using pustular material from milkmaids who had cowpox. And he noticed that if individuals had cowpox, they had symptoms similar to smallpox, but they were much more mild, and they weren't as life-threatening. And so he used this material to vaccinate people so they would develop cowpox and develop a lifelong immunity to smallpox. In 1847, Ignaz Semmelweis demonstrated that childbirth fever could be spread by physicians, and it could be prevented in institutions where children were born using disinfectants from chlorine salt. This was the first time in history where a doctor or a scientist recognized that disease is spread by an organism that is invisible to the eye. And he was able to prevent this disease by proposing the first use of chemicals as to be used as disinfectants. Here's Ignaz. In his birthing chambers, he instructed all of his interns to wash their hands between patients with a solution of chlorine salts. Today we know this as bleach. It was sodium hypochlorite. In his birthing wards, his incidence of childbirth fever or peripheral fever was about 1 or 2%, whereas in the other birthing wards it was 20 or 30%. You would have been amazed to see these doctors in action in 1847. They didn't look like doctors. They looked like they worked in a butcher shop. They wore the same coat, the same clothes from patient to patient. They didn't wash their hands. They would sometimes go to the morgue and perform autopsies and then go up and see a patient, never washing their hands in between. So it's no wonder that diseases like childbirth fever ran rampant in these institutions. In 1858, Virchow declared that all living cells come from existing cells. He reiterated the idea of biogenesis. There is Virchow. But the big breakthrough that destroyed the theory of spontaneous generation came from a French scientist, Louis Pasteur. So when all the other people had failed, and this false theory continued to exist for centuries, how did he manage to pull this off? He did it with this little vessel that you see to the left. So, Pasteur poured broth into specially designed flasks that his glassblower had prepared. They had a long, straight neck, so he could fill them up with broth. And then he had the glassblower bend the neck into this swan neck shape. The end of the swan neck was open to the air, so critics could not claim that air or oxygen could not get inside the flask. His S-shaped flask kept the microbes out, but allowed air to go in. So, by heating this broth, he essentially sterilized it. He would heat it, let it cool, heat it, again let it cool, heat it, and if he did this 2 or 3 times, the broth would remain clear and sterile for months and months and months. Again, the critics could not claim the air could not get inside because the end is open. And what happened to the organisms that normally contaminated Needham's broth was that they were floating around in the atmosphere in the dust, and they would get trapped in the very bottom part of that neck and could not go up the long, slender part above it. And so, therefore, air could get in, but the dust and microbes could not. In 1864, France had a terrible epidemic. A lot of their wines were being contaminated, and instead of having a nice fine wine of alcohol, they were contaminated with an Acinetobacter, and these bacteria turned the wine into vinegar. Well, you can imagine the French as they went to open a nice bottle of wine and it tastes like vinegar. So Pasteur was called in to help resolve this problem. He remembered from the time that he dispelled spontaneous generation using the swan neck flask that he could heat this broth and let it cool, heat it and let it cool, and he called this process pasteurization. He found that if he pasteurized the wine, it didn't become contaminated and the wines remained pure and did not get converted to vinegar. So he became an overnight sensation, not only for his elimination of the theory of spontaneous generation, but also for saving the wine industry in France. Here's Pasteur with his swan neck flask. Once Pasteur dispelled spontaneous generation, the whole world of microbiology tended to open up. And this is known as the Golden Age of microbiology, which started around 1860, 1865, and went to about 1910.